Massive reforestation and rehabilitation programs call for large quantities of seeds and seedlings. A successful program depends on a sustainable supply of good quality seeds and seedlings. Therefore, it is essential to clearly understand the biology of seeds and trees, and the genetic and physiological characteristics of seeds and seedlings. Nursery workers, silviculturists, seed orchard managers, cone collectors, and seed dealers have a very practical need for the knowledge to obtain high-quality seeds and seedlings for forest reforestation and rehabilitation programs. Seeds of tree species are the most common type of forest reproductive material. They are obtained from a variety of sources, including wild stands, selected seed stands, seed orchards, research trials, plantations, and even individual trees on farms, depending on tree species and countries. This module describes the basic principles that cover the biology of forest tree seeds and examines how these principles might apply to reforestation. Angiosperms and gymnosperms have different life cycles. With this knowledge of the life cycles, it is possible to realize the maximum potential of seeds and produce high-quality seedlings. Various treatments based on the physiological requirements of seeds are used to stimulate and enhance germination. Life cycle of angiosperms. Microspores develop into pollen grains, which are the male gametophytes, while megaspores form an ovule that contains the female gametophytes. When the pollen grain reaches the stigma, it extends its pollen tube to enter the ovule and deposits 2 sperm cells in the embryo sac. The 2 available sperm cells allow for double fertilization to occur, which results in a diploid zygote and a triploid cell which acts as a food store. When the fruit falls to the ground, the seed can grow into a seedling, a new diploid sporophyte. Life cycle of gymnosperms. Inside male cones, male spores develop into male gametophytes. Inside female cones, female spores develop into female gametophytes. Pollination occurs when pollen is transferred from a male to a female cone. Sperm then travels from the pollen to an egg, and fertilization occurs. The zygote develops into an embryo within a seed. If the seed germinates, it may grow into a mature sporophyte tree, which repeats the cycle. A seed is a unique package containing the essential structures of a new seedling and the nutrients to support early growth. A fully developed seed consists of an embryo surrounded by nutritive tissue, all of which is enclosed in a protective seed coat. Nutrient tissue provides the energy supplies and raw materials needed by the germinating embryo. Nutritive tissue of broad-leaved tree seeds is called the endosperm and carries a chromosome complement of 3N. In conifers, the nutritive tissue is referred to as the megagametophyte and contains a large proportion of fats and proteins. The embryo contains the genetic makeup of the new seedling. The embryo is the product of fertilization, or the union of the egg from the female parent with the sperm contained in the male pollen. The majority of commercial timber species are wind-pollinated and monoecious. These are trees producing male and female flowers, or strobili, on the same tree. The locations of these flowers usually favors cross-pollination. Some tree species are dioecious, with the sexes separated on different trees. Fortunately, many of these genera can be propagated vegetatively. Seeds come in a variety of shapes, weights, colors, and sizes, depending on the species. Some seeds are in a protective nut like an acorn, while others are in fleshy fruits like the black cherry. The fruit of pines is a cone, and the seed is winged and resembles a miniature helicopter when falling. Seeds develop from male and female parts of the trees producing fruits. Seeds will germinate and grow when they meet favorable conditions. Wind, water, animals, and people disperse seeds to the forest floor, open fields, yards, and roadsides. Only a small number, less than 140, of the world's 50,000 or so tree species are being used in commercial forestry. In many countries, seed production areas do not meet the demand, and seeds are thus collected from poor seed sources such as the wild, untested populations, and scattered trees on farms, increasing risks of failure and low yield. Large-scale multiplication through vegetative techniques such as micropropagation, microcuttings, or somatic embryogenesis is increasingly deployed in forestry to clone superior trees or to propagate new genotypes otherwise available only in limited quantities. However, current efforts of tree breeding still focus on fewer taxa. The gene pools of many tree species, even in breeding programs, are still semi-wild. In contrast, only a few tree species have been domesticated at a similar level to the agricultural crops. Seeds can be collected from good trees, seed stands, seed production areas, and seed orchards. For immediate needs, seeds are produced from a few or limited number of trees, seed stands, and seed production areas that are superior. For long-term and high forest productivity, seed orchard seeds are recommended to use. A seed orchard is a plantation of genetically improved trees, isolated to reduce pollination from genetically inferior outside sources and intensively managed to produce frequent, abundant, and easily harvested seed crops. It is established by setting out clones or seedling progeny of trees selected for the desired characters. Seed orchards are often established while genetic evaluation of the parent material is still underway. In such cases, components shown by progeny testing to be genetically undesirable will subsequently be removed from the seed orchards. The objectives of forming seed stands or seed production areas are to produce seed of improved inherent quality by selecting and favoring seed trees which are vigorous, straight-stemmed, and healthy and produce wood of good quality. Concentrate seed collection into a few specially treated parts of the forest, thus making seed collection easier to organize and control. Improve the germinative energy and germinative capacity of the seed collected. All three can be achieved by one simple treatment, that is, the careful selection of seed trees and the complete isolation of the crowns of these seed trees by thinning. Seed orchards aim to produce high-quality seeds and identify seed origin, while the other seed sources are not for operational planting. Management of seed orchards is most intensive, followed by seed production area, seed stand, and seed tree. Seed quality is most excellent in seed orchards, followed by seed production area, seed stand, and seed tree. Several seed collection methods include using ladders, mechanical lifts, netting, vacuum machines, tree shakers, and helicopters. In general, manual collection by climbing trees is the default way of seed collection. Ladders and ground netting methods are generally considered the cheapest and most practical methods. Well-trained small animals such as monkeys can also be used. Seed testing minimizes the hazards in crop production, determines seed quality, saving time and financial losses. In purity tests, seeds are physically examined to determine the percentage against other debris. Germination testing evaluates the speed at which seeds germinate and their ability to turn into strong and healthy seedlings under a range of environmental and field conditions. Estimating the germination potential of a seed lot takes several weeks to complete, and for some species, pre-treatment may take some additional weeks or months. For this reason, some other methods could be replaced and harmonized. For example, cutting test, tetrazolium test, TTC, excised embryo test, radiographic method, X-ray test, etc. Seed storage is the preservation of viable seeds from the time of collection until they are required for sowing. However, it is essential to store the seed for varying periods, which can be up to 1 year. It is practical when seed production and afforestation are regular annual features. Or 1 to 5 years or more, when a particular tree species bears sufficient seed at several years. Therefore, an adequate amount of seed should be collected in a good seed year. Long-term storage is for conservation of genetic resources. Foresters can produce healthier, more uniform, and vigorous seedlings by using quality seeds. There are mainly 4 practices used in the production of seedlings. Direct sowing of the seeds on site, sometimes with pre-germinated seeds. The use of dip treatment when transplanting bare root seedlings. Before transplanting, sow the seeds in the substrate and soak the plant's root in the fertilizer solution by mixing manure and water. The production of seedlings in nursery pellets. Roots are wrapped with round-shaped pellets containing fertilizer. The use of spontaneous seedlings formed by spreading the seeds of fertile trees. It is possible to transport the spontaneous young seedlings directly with their clods. Seedlings can be produced asexually. Vegetative propagation is any form of asexual reproduction where a new tree grows by cutting a scion off from the parent tree or tissue culture. Cutting is one of the easier ways to make mass propagation. For successful grafts, it is better to use compatible trees. Tissue culture is a method of mass propagation in which fragments of tissue from a tree are transferred to an artificial media where they can continue to survive and turn into new individual trees. For a successful seedling production, it is vital to understand these 4 key aspects. Proper media fill and seed sow. Media or trays should be uniform and contain enough nutrients. Proper moisture. The amount and frequency of watering depends on the species, environmental conditions, and growth stage. Controlled environment. Temperature, light, and shade should be controlled during seed germination and subsequent development. Insect and disease should also be controlled. Plant growth regulator, PGR, application. Success lies in knowing what to apply, at what rate and time. Mycorrhizae could also be used. How can improved seeds be economically produced in sufficient quantities for large-scale forestry programs? This can be achieved by tree breeding, tree improvement, and genetic improvement. Tree breeding produces especially desired products such as trees that are pest-resistant or trees with better quality wood. In tree breeding, we first select 1 or more plus trees from natural stands or plantations. A plus tree shows better performance compared to its neighbor trees in terms of growth, stem and crown characteristics, and resistance to disturbances. Then, artificial crossing is made among the selected plus trees, using techniques such as artificial pollination, stem cutting, grafting, and/or tissue culture. After that, we conduct genetic tests on the plus trees and reselect superior families of trees. The cycle of plus tree selection and artificial crossing is repeated as the generation of trees advance. By means of tree breeding, we can make a valuable forest in the future. For the plus tree selection, ideal traits are fast growth, tree height, diameter at breast height. We want a tree with fast growth, high volume growth at the same time, good wood quality, for example, low lignin content but high cellulose content. For better use of lumber, a tree should be straight and has small branches. Narrow crown. When considering climate changes, it would be valuable to have tolerance or resistance against abiotic and/or biotic stress. For nut or fruit species, it would be better to have high production of flower and fruit production. Tree improvement is the integration of silviculture techniques such as site preparation, fertilization, pesticide use, thinning, etc. during tree breeding cycles. Genetic improvement follows the same cycle as that of tree breeding, but with particular attention to the plus tree's genetic makeup. Genetic improvement has 5 steps. First, target species and provenances are chosen to breed and conserve genetic materials, considering the climate and soil conditions of the plantation area. Second, the amount and cause of variation are surveyed and analyzed using provenance trials and progeny tests. Third, valuable genes are packaged into individuals by seed orchard establishment, gene transformation, etc. Fourth, new varieties and improved seeds are mass propagated. And finally, the breeding population and stock should be maintained as clone bank or clonal archives for future breeding programs. A genetically improved tree comes from a strict selection process where the progeny, offspring, has a carefully chosen trait or a combination of traits for superior tree performance. In short-term tree breeding, plus trees are selected from natural stands or plantations. The selected trees are propagated and used for establishing the first-generation seed orchard and genetic test trials. Genetic information is obtained from the test and transferred to the seed orchard, SO, for genetic thinning. In the seed orchard, genetic gain comes from the selection first and then the genetic thinning a few times. Based on the genetic test, the artificial crossing is made among superior trees and propagated by cuttings, grafting, or tissue culture. And then a generation goes to the next, and so forth. The long-term objectives of a national tree seed program are to ensure a sustained supply of high genetic and physiologically good quality trees for afforestation, and conduct tree planting schemes to create well-adapted populations of woody species. The short-term objectives are to prepare strategies for seed procurement and tree improvement, which will make it possible to cover the immediate seed demand, and incorporate long-term measures such as selection, conservation, establishment of seed sources, and breeding populations. Seed procurement is to produce seeds of high physiological quality for a sustained supply of plants. Tree improvement is to produce genetically more desirable trees, including tree breeding. Gene resource conservation is to protect and maintain genetic variation for sustained utilization. Life is death. Death is life. Form and no form are the same. Seed is disappearance of the tree. The tree has moved into no form. Nothingness has tremendous potential. One single seed can fill the planet with greenery. Seed is a carbon sink, and seed production is carbon capture.